Dear students, we'll discuss briefly on the topic morphological, biochemical and DNA-based markers, namely RFLP, AFLP, RAPD, SSR and DNA probes. We'll discuss this topic with reference to agricultural sciences. Biochemical and molecular techniques have shortened the duration of breathing programs from years to months, weeks, or eliminated the need for them altogether. The use of molecular markers in conventional breathing techniques has also improved the accuracy of crosses and allowed breathers to produce strains with combined traits that were impossible before the advent of DNA technology. This breathing technique can be used in almost any species, whether animal, plant, fungi or bacteria, but the focus here will be on marker-assisted selection in agriculture. Various markers used in plant breeding are morphological markers, biochemical markers, molecular markers. We'll discuss the various markers used in plant breeding one by one. First, morphological markers. Morphological mutant traits in a population are mapped and linkage to a desirable or undesirable trait is determined and indirect selection is carried out using the physically identifiable mutant for the trait. There are several undesirable factors that are associated with morphological markers. The first is their high dependency on environmental factors. Often the conditions that a plant is grown in can influence the expression of these markers and lead to false determination. Second, these mutant traits often have undesirable features such as dwarfism or albinism. And lastly, performing breeding experiments with these markers is time-consuming, labor-intensive, and the large populations of plants required large plots of land and greenhouse space in which to be grown. Next marker is biochemical markers. Isoenzymes are used in biochemical markers in plant breeding. Isoenzymes are common enzymes expressed in the cell of plants. The enzymes are extracted and run on denaturing electrophoresis gels. The denaturing component in the gels, usually SDS, unravels the secondary and tertiary structure of the enzymes, and they are then separated on the basis of net charge and mass. Polymorphic differences occur on the amino acid level, allowing singular peptide polymorphism to be detected and utilized as polymorphic biochemical marker. Biochemical markers are superior to morphological markers in that they are generally independent of environmental growth conditions. The only problem with isoenzymes in marker-assisted selection is that most cultivars, that is commercially breeds of plants, are genetically very similar and isoenzymes do not produce a great amount of polymorphism. Polymorphism in the protein primary structure may still cause an alteration in protein function or expression. We'll discuss on molecular markers. Molecular markers are based on naturally occurring polymorphisms in DNA sequences. There are various methods to detect and amplify these polymorphisms so that they can be used for breeding analysis. Molecular markers are superior to other forms of marker-assisted selection because they are relatively simple to detect, abundant throughout the genome, even in the highly bred cultivars, completely independent of environmental conditions, and can be detected virtually at any stage of plant development. Molecular markers can be used for several different applications, including germplasm characterization, genetic diagnostics, characterization of transformants, study of genome organization, and phylogenetic analysis. Now we'll come to the various molecular marker techniques. First, we'll see the restriction fragment length polymorphism, or which is known as AFLP. This was the first molecular marker technique developed and used in marker-assisted selection for plant breeding. The technique centers around the digestion of genomic DNA digested with restriction enzymes. These enzymes are isolated from bacteria and consistently cut DNA at specific base pair sequences, which are called recognition sites. These recognition sites are not associated with any type of gene and are distributed randomly throughout the genome. When the genomic DNA is digested with one of these restriction enzymes, of which there are thousands each cutting at specific sequence, a series of fragments are produced of varying length. These fragments are separated using agarose or polyacrylamide gel electrophoresis, or known as PAGE, and yield a characteristic pattern. DNA has a uniform charge per unit length when run under electrophoresis conditions, which arises from the phosphate groups in its backbone. So when DNA fragments are separated via electrophoresis, the distance they travel is dependent only on their molecular weight. This allows their molecular weight to be determined with simple standard called DNA ladders, which are run alongside the DNA in the gel. When restriction fragments are separated on agarose gel, a series of bands results. Each band corresponds to a restriction fragment of different length. The lighter they are, the farther they have traveled. Variation in the characteristic patterns of a RFLP digest can be caused by base pair deletions, mutations, inversions, translocations, and transpositions, which result in the loss or gain of a recognition site, resulting in a fragment of different length and polymorphism. Only a single base pair difference in the recognition site will cause the restriction enzyme not to cut. If the base pair mutation is present in one chromosome but not the other, both fragment bands will be present on the gel, and the sample is said to be heterozygous for the marker. Only codominant markers exhibit this behavior, which is highly desirable. Dominant markers exhibit a present or absent behavior, which can limit data available for analysis. Now let's come to the procedure for RFLP. The first step is DNA isolation. a significant amount of DNA must be isolated from the sample and purified to a fairly stringent degree, as contaminants can often interfere with the restriction enzyme and inhibit its ability to digest the DNA. The next step should be restriction digest. Restriction enzyme is added to purified genomic DNA under buffered conditions. The enzyme cuts at recognition sites throughout the genome and leaves behind hundreds of thousands of fragments. After this, the next step will be gel electrophoresis. The digest is run on a gel, and when visualized, appears a smear because of the large number of fragments. The next step is transfer to nitrocellulose membrane filter. And the next step is probe visualization. Because of the large number of fragments, probe must be constructed to visualize more specific bands in the digest. These probes consist of radio-leveled oligonucleotide sequences which will anneal to the fragment sequences so that they may be visualized on photographic paper using a technique called a Southern blot. Next is the analysis. To develop a probe to screen AFLP, an initial digest must be performed on the species of interest. The restriction fragments are ligated into a plasmid vector and transformed into bacteria. Positive cultures are then isolated, ligated sequences removed, amplified by speciar and radio-leveled for use as AFLP probes. Next, we'll come to the PCR-based molecular markers. The first one is the randomly amplified polymorphic DNA markers known as RAPD. RAPD are produced by repeated cycles of DNA denaturation, renaturation, DNA replication in a PCR, that is polymerase chain reaction equipment. The genomic DNA of the selected strain is mixed with an excess of a selected oligonucleotide, a short polynucleotide, and this mixture is then used for DNA amplification in a PCR. The segment of genomic DNA amplified or replicated repeatedly will be continuous to that segment which is homologous to the oligonucleotide used. A distinct band will be seen on the agarose gel, while this band will be absent in other strands. Thus, RAPD's bands are either present or absent in a strain, and the F1s generally express the bands of both the parents' codominant expression. RAPD analysis is facilitated by the availability of base sequence of a short segment of genomic DNA. Variation in this segment can now be detected by generating an oligonucleotide that is homologous to it. But oligonucleotide having any base sequence can be used in these studies. The oligonucleotides can be prepared by using an oligonucleotide sensitizer. The information obtained from RAPDs can be used for linkage mapping much in the same manner as that from RFLPs. It is important that the RAPD's approach does not utilize restriction enzymes, which are very costly, and probes of any kind. This saps the effort needed for developing suitable probes and the risks of handling radio-labeled materials. It utilizes in the PCR a DNA polymerase which is resistant to the high temperatures needed for DNA denaturation, so that it is reused repeatedly during DNA amplification. RAPD was the first PCR-based molecular marker technique developed, and it is by far the simplest. Short PCR primers, approximately 10 bases, are randomly and arbitrarily selected to amplify random DNA segments throughout the genome. The resulting amplification product is generated at the region flanking a part of the 10 base pair priming sites in the appropriate orientation. RAPD often shows a dominant relationship due to primer being unable to bind, showing 3:1 ratios, unable to distinguish between homozygotes and heterozygotes. RAPD products are usually visualized on agarose gel stained with ethidium bromide. Next, we will come to the next marker, simple sequence repeats, or known as SSR, or microsatellites. These are short DNA fragments of usually less than 100 base pair and are made up of tandem repeats of 2 to 7 base pair. Simple sequence repeats are present in the genomes of all eukaryotes and consist of several repeats of 1 to 4 nucleotide motif. Some common motifs are mono, say A or T, di, maybe AT or GA, or tri, AGG, or maybe tetra, AAAC. These repeated motifs are denoted as AAAC and, where and is the number of tandem repeats. The sequences flanking these microsatellites are often conserved and can be used to design primers. These primers can be designed by constructing a novel genomic library and sequencing segments of the subject genome. Already discovered sequence, that is GenBank online database, can also be searched for SSRs and primers designed from that. Polymorphism is based on the number of tandem repeats and therefore the length of the PCR products. SSR is a codominant marker such as AFLP and is usually visualized on metaphor agarose or polyacrylamide gels. The next marker will be amplified fragment length polymorphism, or AFLP. AFLP is the latest form of marker-assisted selection and is highly sensitive method based on the combined concept of RFLP and RAPD. This technique is applicable to all species, giving very reproducible results. The basis of AFLP is the PCR amplification of restriction enzyme fragments of genomic DNA. DNA is cut with two specific restriction enzymes, one frequent cutter, 3 base pair recognition site, and one rare cutter, 6 base pair recognition site. Oligonucleotide adapters are ligated to the ends of each fragment, one end with a complementary sequence for the rare cutter and the other with the complementary sequence for the frequent cutter. This way, only fragments which have been cut by the frequent cutter and rare cutter will be amplified. Primers are designed from the known sequence of the adapter plus 1 to 3 selective nucleotides, which extend into the fragment sequence. Sequences not matching these selective nucleotides in the primer will not be amplified. Next, the PCR is to be performed. The next step is to visualize on agarose gel with ethidium bromide. Typical results give 50 to 100 bands despite selective nucleotides and rare or frequent selection. This high number of bands eases analysis by providing more chance of polymorphism. Now we'll come to the DNA probes. DNA probes are small segments of DNA which help to detect the presence of a gene of a long DNA sequence in a biological system. These DNA probes are prepared for commercial purposes and are believed to be the most sophisticated and sensitive means to identify genes or specific DNA sequences. DNA probes provide commercial avenues for diagnosis of disease infections, identification of food contaminants, for isolation of genes, and in other microbiological tests. It is believed that these DNA probe assays for a variety of purposes will be cleaner, simpler, faster, and cheaper than the traditional microbiological test, and are also expected to be 100-fold more sensitive. The production of DNA probes can be done by any of the following methods, such as using a template DNA with the help of purified biological enzymes, or DNA probe of specific sequence can also be obtained by using automated DNA synthesizers, or DNA probe can also be included in viral DNA and maybe even multiply in bacteria. Thus, by this way, many copies of DNA probe can be obtained. Now, let's come to the conclusion. Conventional breeding methodologies have extensively proven successful in development of cultivars and germplasm. However, subjective evaluation and empirical selection still play a considerable role in conventional breeding. Marker-assisted breeding has brought great challenges, opportunities, and prospects for conventional breeding. As a new member of the whole family of plant breeding, however, marker-assisted breeding as transgenic breeding or genetic manipulation thus cannot replace conventional breeding, but is and only is a supplementary addition to conventional breeding.